our discussion of Chapter 10, Microbial Growth. And what we're going to do then is do follow-up, not follow-up, we're going to do Part 2, which is our discussion of selective and differential media. So a reference here is going to be Table 2 on page 90. Now, one of the slight issues was I thought we were going to have McConkey's media, but we don't. We have Eosin methylene blue media, and I know I asked you guys to prepare the McConkey's. I asked you to read about McConkey's media. So on the lab practical, there'll be some bonus questions on McConkey's media, so you don't feel frustrated like you studied it for nothing. And what I'll do is I'll first tackle this Eosin methylene blue media because it's one of the more complicated media that we're going to work with. Okay, so Chapter 10, and this is Part 2, Selective and Differential Media. So first of all, we're going to tackle what we mean by selective media. And this, I think that the vocabulary here can get pretty confusing. So if we ask ourselves, what do we mean by selective media? What we're trying to do is we're going to permit or select or growth of one type of microbe while inhibiting the growth of other microbes. And I know it's like, what the heck does that mean? But let's first make sure we have all the vocabulary under our belt. Okay, so when you see, when we use the term select, we're going to select for this one group of microbes. That means we're going to permit them to grow while inhibiting or stopping the growth of all other microbes. So select and inhibit, you can think of them as opposite. They're opposite in meaning. Now the way most selective media work is that, okay, so we'll see, how are we going to select for a specific group of microbes? The way most selective media works is we add inhibitors in the media that will stop the growth of all the microbes we're not interested in. Okay? So how we're going to add inhibitors to the media? That's all right. We're going to add inhibitors to media to stop, and again, stop means to inhibit the growth of the undesired microbes. Okay, and probably the easiest way to hopefully make this understandable, let's use a specific example. Okay, so for example, okay, we're going to pretend we're public health microbiologists. And one of the important jobs of public health microbiologists is to make sure that drinking water is safe. Okay? And the biggest fear we have as public health microbiologists is that raw sewage or feces would contaminate drinking water. And why are we worried about that? Well, we know there could be all kinds of pathogens in feces. There could be the worm eggs, like the ascaris eggs, the Tania eggs. There could be Giardia, Valentidium, Entamoeba histolytica could be in feces. We could have bacterial pathogens like Salmonella, Shigella, toxin-producing E. coli, Vibrio cholera. Okay. So there's so many different pathogens that are present in feces, right? So that's why we don't want feces in our drinking water. It's not just the OO value, it's the fact that so many pathogens are present in feces. So this is a huge issue. Oh, poliovirus, hepatitis A virus, right? So we could ask ourselves, well, gosh, would we screen for all those different pathogens? I mean, it would be overwhelming, right? So what microbiologists have done is they've said, well, let's do this. Let's choose an organism that's really easy to grow in the lab, will grow overnight, doesn't take a lot of fancy equipment or fancy media to grow it, and we'll use that organism as an indicator of fecal contamination. Okay, so we talk about indicator organisms of fecal contamination. Okay, so if we see a lot of these indicator organisms present in drinking water, we're like, uh-oh, this means feces could be present, raw sewage could be present, and that means any of those other pathogens could be present. Okay? So historically, our indicator of fecal contamination has been good old E. coli. Historically, E. coli has been used as the indicator of fecal contamination. And the reason is for E. coli to flourish, usually it has to be living in the intestinal tract of an animal. Okay, so so if we find lots and lots of E. coli present in drinking water or on food, it suggests the drinking water of the food's been contaminated with feces. Okay. Now another organism that we'll talk about a little bit later, and more recent years has been used as an indicator, is a second fecal indicator that we'll just put here in parentheses and come back to, is enterococcus. Okay, so we'll just put that there and we'll come back to that later. We're going to first focus on E. coli. So if we think, let's take an example that we're testing wells out in the countryside, and we're like, we want to make sure the well water is pure or not fecal contaminated. Well, one of the problems is when you're taking these samples, you might have other microbes present. For example, there could be just good old free-living soil microbes present in the well water, not organisms associated with fecal contamination, just organisms living out in the dirt. So what we want to do if we're taking well water samples is we want to inhibit the growth of all but the organisms that would suggest fecal contamination. Okay? So we want to develop that. So E. coli belongs to this big family of bacteria called Enterobacteriaceae. And there's a little description in chapter 10 of this big family of Enterobacteriaceae on page 91. Okay? And also, if you want a little bit more, at the start of the food microbiology, food water microbiology, where is that? Where'd that go? Okay, chapter 12, which starts on page 104, there's a further description of some of these fecal indicator organisms, just for those of you that are interested. Okay, all right. So, if we're public health microbiologists, we want to screen these well water samples for possible fecal contamination, and we're going to use E. coli as our indicator organism. What we'd like to do is use a media that will inhibit all other microbes and permit or select for the growth of this big family Enterobacteriaceae. That's one way for us to make our work a lot easier, right? Okay, so we're going to then select for this group Enterobacteriaceae. And these are often referred to, they're often nicknamed as enterics. And enteric refers to the intestine. Okay, so enteric bacteria are those that can live in the intestinal tract. So we want to select for enterics, we want to let them grow, and we want to inhibit all the other microbes, all the other bacteria that might be present in that well water. Okay? So what we're going to do then is use a selective media. And what we want to do is select for those enterics or Enterobacteriaceae. And we want to inhibit all other microbes. And we want to inhibit all other microbes. All right, so which kind of selective media can we use? Well, the one we have for you today is called Eosin methylene blue medium, or agar. And Eosin methylene blue on table 2 on page 90, it's the one to the third medium down. Okay? So we're just going to take a look at what is in Eosin methylene blue that will select for the growth of those enterics and inhibit all the other microbes. So our specific example is Eosin methylene blue. And you guys know we are notorious for using initials, so often you just say EMB agar. Now, we want to know what are the inhibitors in Eosin methylene blue. What are the substances present that will inhibit all but the enteric bacteria? So can you guys help me out here? By looking at the second column over, you'll see the second column says inhibitors or selective ingredients. What are the inhibitors of all but the enteric bacteria that's present in EMB? Eosin Y and methylene blue. Good. Okay. So the inhibitors are these dyes called Eosin and methylene blue. Okay. Now the next thing we want to ask is, so what does Eosin and methylene blue inhibit? And that's the third column over. Which microbes will be inhibited from growing?
Gram-positive bacteria.
Gram-positive bacteria and non-enteric gram-negatives. Good. Okay, good. Okay, so these guys will inhibit, and that needs to stop. They inhibit gram-positive bacteria and non-enteric, so meaning they don't live in the intestine, gram-negative bacteria. So hopefully, ideally, only enteric bacteria will grow, and that's exactly what we're after, right? Okay, so the fourth column over asks us what is selected for. So EMB selects for the growth of which? So who will be selected for? Who do we want to grow? The enterics, those Enterobacteriaceae, great. And I know you guys, this is just like, oh my goodness, this is so painful. But the reason we're going over this in some detail is what we see on the lab practical, the next lab practical, this information is going to be on. People get a lot of points wrong because I don't think they understand the vocabulary. So that's why we're trying to use these in terms, you know, selects, inhibits, inhibitors, what is inhibited, what do we select for? Because usually in the lab practical, I think people just don't understand the terminology, and that's why they get it wrong. So that's why we're doing this kind of stuff. Good, okay. So if we took our well water sample, and let's presume that there's a mixture of, let's say there is fecal contamination, so we have enteric bacteria present in the well water, but maybe some, in addition, some free-living bacteria from the soil. Okay? So if we plate our water sample, say this is the EMB agar, and I'm just going to make this up, you guys. Let's say the Enterobacteriaceae bacteria all put in orange, okay? And you know, obviously they're making them much bigger than they should be, okay? So we'll put it, the enterics will be in orange. And let's say the non-enterics will be in white. Okay, and there's a mixture of both of them in a well water sample. Okay, so we spread it out over the EMB agar, we incubate it. Which one of these bacteria will grow on the Eosin methylene blue? The enteric bacteria, right? And who will be inhibited? The non-enterics, right? So following incubation, those non-enterics, they won't grow, right? They're inhibited, they're stopped from growing by the Eosin and methylene blue. So after incubation, when we pull those plates out, we should only see colonies of what?
Enteric.
Yeah, we selected for the growth of those enterics. So we would expect that the colonies should be colonies of the enteric bacteria, because all the other free-living bacteria, they were inhibited. They weren't permitted to grow. Okay. So that's this concept of selecting for the growth of one group of bacteria by inhibiting all the others we're not interested in. Okay, and it makes screening samples, you know, food, water samples, much faster, easier. Okay. So now it even gets more complicated. Now it gets more complicated because it turns out that EMB is not only selective. It's what's called differential media. It lets us distinguish between different types of microbes amongst those that do grow. This gets a little bit wild. hang in there with me here. Okay, so, we said we're talking about selective and differential media. So it turns out that Eosin methylene blue is also an example of what's called a differential media. And differential media permits us to distinguish or differentiate between different types of microbes that do grow. So in our specific example of our selection for this big family Enterobacteriaceae, there's many different types of bacteria in this big family. So we want to differentiate between the different groups. And the way we're going to differentiate them is through their ability to ferment lactose. Differentiate by ability to ferment lactose. And I just want to tell you all, this information we're going to go over, we're going to see it again. So don't despair that, oh my gosh, all this information, we learned it for one lab and then that's it, because we're going to see this over and over again. So it will be worth it to learn it. Okay, so what we do, the big question we're going to ask here is, can lactose be fermented? That's the question we're using to differentiate between all the different groups of Enterobacteriaceae. Okay, so we'll do a split here, and we'll say, over here, yes, lactose can be fermented. Over here, no lactose fermentation. Okay, and we often use the abbreviation lac negative. When an organism can't ferment lactose, we use the abbreviation lac negative to say it can't ferment lactose. So let's just ask ourselves, if some of these enteric bacteria, if they couldn't ferment lactose, okay, it turns out that lactose is the only sugar present in Levine's Eosin methylene blue agar. So if we have enterics that can't ferment lactose, they have to tear apart the proteins instead. They use proteins as a source of carbon and energy. And when they tear the proteins apart down into amino acids, and they tear the amino acids apart, they release the amino groups as ammonia. And you guys might remember from chem that ammonia acts as a weak base. It can bind to hydrogen ions. And consequently, the hydrogen ion concentration goes down. What happens to pH? Okay, it goes up, right? So what we'll have, if we have lactose negative enterics growing on Eosin methylene blue, we'll have an increase in pH, and we'll see no color change in the medium. The colonies, the agar remains the same color as it started out as, okay? So that's not very exciting. Okay. However, there are some really cool lactose negative pathogens. For example, salmonella is lac negative. And another important bacterial pathogen is Shigella dysentery, that causes bacterial dysentery. Those are both lactose negative enterics. Okay, so they can grow on Eosin methylene blue, okay, but they won't have any color change in the media, nor their colonies will appear basically the same color as the media. So that's not very exciting, right? Okay, where it gets kind of cool is over here with members of Enterobacteriaceae that can ferment lactose. And here's where it gets a little bit complicated. It turns out different microbes have different fermentation pathways. Remember that from lecture? Mm-hmm. Okay, different microbes can make different enzymes and have different fermentation pathways. So one of the fermentation pathways is called mixed acid fermentation. And it's called mixed acid because end products include a great variety of acids. Okay, and as a consequence, the final pH is very low, less than pH 4.4. Okay, and at this really low pH, it turns out the Eosin and methylene blue will precipitate Okay, so we get precipitation of the Eosin and methylene blue. And as a consequence, we get dark colonies. And what's really cool is the surface of the colonies have this metallic green sheen from the precipitated Eosin and methylene blue. Dark colonies with metallic green sheen. Okay, now, why don't I, before we keep going, let me pass out some of these plates that hopefully, I think we're starting to lose our metallic green sheen. That's a shame. Okay, let me get this other set of plates. The microbe that we classically think of as carrying out this mixed acid fermentation is our fecal indicator, E. coli. Okay, so remember, this is our indicator of fecal contamination. So if we plated well water samples on Eosin methylene blue and incubated them, and when we pulled them out of the incubator the next day, they were covered in these dark metallic green colonies, we're gonna, well, we won't panic, right? We're professionals, right? But we're like, oh no, you know, this well water is potentially contaminated with feces, and we need to tell the families using it, stop using it, right? Or boil it, you know. So let me just pass some of these demo plates out, and let me describe what's on them. So the Eosin methylene blue that has a single organism on it, this is E. coli, and sadly what happens is when E. coli is permitted to grow on these plates for a long period of time, they use up all the lactose and then they start attacking the amino acids. So we get a shift from a very acidic pH to an alkaline pH, and we start losing that metallic green sheen on the surface. But what you want to do is just shine the light on the surface of the colonies and see if there's a little hint of a metallic green to the colonies. Okay? And you also want to notice that the colonies themselves are dark. They have a dark appearance. Okay? So let me just pass these out so you all have something to look at. And we'll probably have to end up sharing. Okay?